Hello, in this video we will discuss about the purine metabolism. So what is purine? Purine is the double ring structure of the nitrogenous base in the DNA, also the GTP and ATP. So here the purine are biologically synthesized as nucleotides and in particular as ribotides bases attached to ribose 5-phosphate. So the ribose 5-phosphate is the backbone for the attachment of the bases for synthesis of the bases. So that is the major function. But both adenine and guanine are derived from the nucleotide inosine monophosphate. So the inosine monophosphate derivatives. So what is inosine monophosphate? So we will also understand about the inosine monophosphate. So let's begin to understand. First of all, I want to share the introduction of the purine. So purine is a heterocyclic aromatic organic compound. So the purine is a heterocyclic. Hetero mean the different compound rather than the carbon containing compound. Heterocyclic aromatic ring-like organic compound that consists of two rings. So the two rings fused together, it is water soluble. So you should know about that. And the guanosine triphosphate and ATP, also the DNA is the major compound for the purine. Purine is the double ring structure and it is the adenine and guanine. That is the two type of the nitrogenous base and both are the purine type. While the cytosine, thymine and cytosine is basically is the single ring pyrimidine. So the biosynthesis, if we will discuss, so let's begin to understand the whole detail for the biosynthesis of the purine. So the purine are biologically synthesized as nucleotides and particular as ribotides. Base is attached to ribose 5-phosphate. So the ribose 5-phosphate attachment. So let's draw the picture for the understanding with the ribose 5-phosphate. So the ribose 5-phosphate here I am drawing to understand. So the ribose 5-phosphate means the 5-phosphate means the carbon number 5 contain phosphate group. While here is the basically the 5-phospho alpha D riboseal 1 pyrophosphate phosphate synthase enzyme, the PRP synthase enzyme. This PRP synthase enzyme 1 is used in the presence of magnesium, the coenzyme, by the using of ATP to release the 2-phosphate group, and this 2-phosphate group with attached with the carbon number 1 and carbon number 6 contain phosphate group to form a 5-phospho alpha D riboseal 1 pyrophosphate. So the 5-phospho alpha riboseal 1 pyrophosphate formation. Anyhow, let's begin to understand. Here is the Amido phosphoribosyl transferase enzyme. This enzyme is the PRA transferase, also we can say that is the transferase. So the PRA transferase, by the using of the glutamate, by the adding water to donate the amine group in the presence of a magnesium to produce the 5-phosphate ribotide amine. So it means the carbon number 1 contain amine group while the 5 carbon containing the ribose contain a phosphate group, and in this way you can see in this diagram to understand as well. So let's draw our the other picture. The basically is the phosphoribosyl amine glycine ligase enzyme. In this way you can see this the magnesium is used and ATP and glycine to convert the ADP and the and the inorganic phosphate to produce the basically is the glycinamide ribonucleotide formation. Why this glycinamide ribonucleotide? Because the this glycine addition, and in this way the glycine addition will lead to produce the glycinamide ribonucleotide, and in this way you can see the GAR we can understand as well. But the next step is the basically is the fourth step is the basically the GAR transferase enzyme used to basically the GAR transferase E2 enzyme 2, which that means it will be trihydrofolate used to formyl trihydrofolate formation in the N10 nitrogen 10, and in this way the phosphoribosyl glycinamide formyl transferase enzyme used and using to produce the formyl glycinamide ribonucleotide, and this formyl glycinamide ribonucleotide formation, and in this way you can see the FGAR. This FGAR means the formyl glycinamide ribonucleotide formation, and in this way here you can see the water and ATP and glutamine will use to in the presence of the FAS formation to form the releasing of the glutamine to ADP also to produce the in the presence of the phosphoribosyl formyl glycinamide synthase enzyme to synthase the phosphoribosyl formyl glycinamide synthase enzyme. This enzyme used to produce the formyl glycinamide ribonucleotide, and this formyl glycinamide ribonucleotide formation. And in this way, the basically here you can the formyl glycinamide ribonucleotide formation, and in this way the FGAM will produce, and in this way the GAR transferase enzyme used in the presence of the ATP to produce ADP, and in this way here you can see this the formation is the air synthase enzyme used, and after this air synthase enzyme used to produce the aminoamidazole ribotide. So aminoamidazole ribotide, and in this presence of ATP and hydrogen bicarbonate to produce ATP, and this phosphoribosyl aminoamidazole succinocarboxyamine synthase enzyme. This enzyme used to produce this structure is known as the carboxamide amidazole ribotides, and this ribotide it is also known as the care, and you can say and understand this is the care. And after this is here is the adenylosuccinylase enzyme used to aspartate in the presence of ATP to produce the normal succinyl and succinyl carboxamide. So the carboxamide formation to produce this structure, and in this way here you can see that care to produce the adenylosuccinylase enzyme to produce this type, and this is the basically that second ring formation, and in this way here you can see the aminoamidazole and succinyl carboxyamide ribotide, and this ribotide formation. In this way aminoamidazole ribotide, you can see aminoamidazole ribotide, so the aminoamidazole, and in this way after this formation basically is here is the succinyl, you can say the succinyl aminoamidazole carboxyamine carboxyamine ribotide formation. To form this, the fumarate will be separate, and here is the adenylosuccinylase enzyme will produce the fumarate, and this fumarate is basically the citric acid cycle is used this fumarate, and in this way they produce ATP, NADH and FADH2. And after this here is formation is the ACAR. It is also known as the 5-aminoamidazole 5-carboxamide ribonucleotide formation, and here is a 5-aminoamidazole 5-carboxamide ribonucleotide formation, and in this way here you can see the N10 formyl trihydrofolate again used to produce the trihydrofolate, and in this way here is the ACAT synthetase enzyme, AICAT synthetase enzyme 1, which that is used to produce the FACAT. In this way the 5-form amidoimidazole 4-carboxamide ribotide formation, and in this way the 5-form amidomidazole 4-carboxamide ribotide formation to produce this type of, and release the water and after the releasing of the water due to the inosine monophosphate synthase enzyme use. At last, here is the enzyme 2, ATIC2 enzyme 2. In this way, here is the inosine monophosphate synthase enzyme to produce the inosine monophosphate. And this inosine monophosphate you can see and understand as well. Here is the IMP. So in this way, here is the monophosphate. It means the carbon number 5 contain phosphate, and in this way monophosphate, if the two phosphate will be attached, is known as the IDP, while the ITP is also produced, the inosine triphosphate. But in this case here you can see the NAD plus used in the presence of water to dehydrogenation, and here is the dehydrogenase enzyme used with the IMP, dehydrogenase enzyme used to produce the XMP. So the XMP mean the phosphate group, basically the monophosphate. In this way the X mean it will be come from the IMP. And after this, here you can see this is the GMP synthetase enzyme used to in the presence of ATP and glutamine and water to release the AMP and pyrophosphate and glutamate. So in this way you can see and understand as well, and basically here you can understand this is the GMP formation, and this GMP formation mean the it is the guanosine monophosphate formation. So this guanosine monophosphate formation due to the it is the guanine formation, and this is the guanosine monophosphate formation. After this, here you can see this is the adenylosuccinate and synthetase enzyme used to produce the adenylosuccinate formation, and this adenylosuccinate formation in the presence of the GTP and the aspartate, that is the amino acid will bind with the amine group, and here you can see, and this is the aspartate, and here is you can see adenylosuccinate enzyme formation. And in this way, here is the fumarate formation, and this is the fumarate and adenylosuccinate lyase enzyme used. After this, adenylosuccinate lyase enzyme used to produce the release the fumarate, but adenosine monophosphate formation. This adenosine monophosphate is a precursor of ATP. So this ATP is also produced, and GTP is also produced from the guanosine monophosphate, will precursor of the guanosine triphosphate and diphosphate, and ATP and ADP and GDP and GMP and GTP. So these all things come from the inosine monophosphate. That is the all biosynthesis pathway we are discussing in this video. So I hope you are getting this concept. That is basically is you can see the whole picture to understand as well. So here is the 5' nucleotidase enzyme used to produce the guanosine, while here the xanthosine formation, and in this way the xanthosine, and here this is the basically the purine nucleoside phosphorylase enzyme used to produce the guanine, and this the guanine is the basically synthesis and separate, and in this way the deaminase enzyme used to produce the xanthine, and the xanthosine is also convert into the xanthine. This is the uric acid is the end product if the not useful, and excess of the guanosine and excess of the purine will excrete as a uric acid to as a waste from the urine. So there are many birds which that involve this process to produce more uric acid, but in the human is less. So all these things is about the purine metabolism and ATP and GTP.